Welcome to day 2 of the Children's Health Workshop, sponsored by a whole bunch of people that you can see on the first page. So today I just wanted to go through a really fast recap of yesterday because there was so much incredible information. A lot of creative and productive work has happened on exposures to environmental chemicals in California by the Children's Health Centers and also in Washington, with some focus on organophosphate pesticides and the difficulties using urinary metabolites to estimate exposure to specific OPs, the complications that has for cumulative risk assessment and so forth. And we heard some very interesting work on dust and what chemicals are in dust and how you go about assessing potential exposures through that pathway. We heard about a navigation guide that's using basically a clinical model to go take a systematic look at information on environmental chemicals and decide whether there's a hazard and what kind of messaging should come from that and what kind of action. And we heard a lot of discussion about getting physicians and other healthcare providers involved in environmental health, and we need a cheering section on the regulatory side, I can tell you that. So today, we're going to start out with a neurodevelopmental talk from one of the children's centers at UC Davis. So I would like to introduce Dr. Isaac Pessa. Isaac is a professor of toxicology, and he's chair of the Department of Molecular Biosciences at UC Davis. He is involved in the Mind Institute with a number of great scientists there. He has a PhD in toxicology from University of Maryland, and then he did a postdoc at UC Berkeley where he discovered the ryanodine receptors. So those are a pretty important class of chemicals for receptors, for neurodevelopment. He's been looking at the molecular biology of those receptors and their role in development. And in particular, for us toxicologists, he's been looking at environmental chemicals, how they influence neurodevelopment, primarily through the ryanodine receptor. Okay, so Isaac is going to give a talk today on tipping the balance of autism risks, exposures that influence neuroexcitability, and oxidative stress.
What I would like to do is talk in more general terms about the accomplishments of the UC Davis Center for Children's Environmental Health, which is now in its 11th year. That is 10 years of funding and 1 year of no-cost extension because we missed the cycle for RFAs. Nevertheless, the Children's Center has been very busy. And so many of you probably know this, but I'm going to go through it quickly, that autism is now estimated to affect every 1 in 74 males and every 1 in about 110 children. It's a severe and pervasive impairment in the development of social reciprocity. It's associated with impairments in either verbal or nonverbal communication skills. Often there are stereotyped behaviors, but they can be of various severities. Restricted interests and activities. In about 30 to 35%, this value here has actually gone up. When more careful diagnosis is done, about 30 to 35% of the kids seem to develop normally for about the first 24 months of age, and then begin a downward slide called regression. There are several comorbidities, and this is where now we're starting to talk about autisms and not autism. That autism is really based on a behavioral diagnosis, a set of behavioral diagnoses. And in doing so, you're probably bringing in, this is a figure that I heard last night from Evan Eichler, who's a geneticist at the University of Washington, 800 genes are most likely contributing to autisms. They're not all in the same child. And so autism is very complicated. But along with the core symptoms are cognitive deficits anxiety disorders, seizure disorders, sleep disorders, and we now know that many of them are prone to immunological and GI problems. A clue into the etiology may be the fact that there's 4-to-1 male to female ratio. So it's a male bias disorder. When I started in this field, I would say 90% concordance in monozygotic twins, less than 10% in dizygotic twins mean that it's the most heritable disorder on the planet. In other words, it's all genes, no environment. Of course, if one took a close look at the studies on which these statements are made, they were really not that accurate. Oops, let's keep going here. July 4th of last year, this paper hit the presses from a group of geneticists as well as some epidemiologists, including Judy Grether, Joachim Hallmeyer. From our place, it was Sally Ozenoff. Essentially, they turned that view around, that autism actually has a concordance in monozygotic twins of less than 60%, and dizygotic twins upward of 40%. So in other words, shared environment is a huge factor in obtaining a autism diagnosis. So this is the new view of autisms, maybe up to the minute, but it's ever changing. Less than 60% concordance in monozygotic twins, more than 30% in dizygotic twins. What causes autism? Well, we know that syndromic forms of autism, in fact, we know of single gene causes of autism. They're very rare. Fragile X, not all that rare. About 20% of these kids do get diagnosed with autism. So that's an expansion repeat in the FMR1 gene on the X chromosome. Rett syndrome, again, MECP2 deletion or mutation, which is a gene methylation protein, so very important in epigenetic modification of the genome, etc. Well, these are disorders that contribute cognitive dysfunction to the kids. One of the outcomes, in fact, may be autism-like symptoms. And so this may not be pure autism. One example of pure autism comes from a very strange sort of gene. It's actually the gene that runs your heart. It's a calcium channel, CAV1.2. This calcium channel is actually the initiator of EC coupling, a cardiac contraction. It's also widely expressed in the nervous system. Kids that have a single missense mutation in this gene, their calcium channels fail to inactivate rapidly enough. They are prone to long QT syndrome. But in fact, they have about a 70% rate of full autism. And so this is one of the clearest links between a single gene defect and autism. We now have evidence that the mouse that's made to have Timothy syndrome has social deficits. And so calcium dysregulation probably is a common convergent pathway that can contribute to autism. But most cases of autism are idiopathic. We really don't know what how they originate. Copy number variations are certainly contributing to autism. Some are inherited from paternal or maternal lineage, but others are de novo. In other words, they don't occur in either parent, biological parent, but they seem to emerge in the child that's affected. And of course, there's evidence now that epigenetic changes are also associated with autism. So the genetics of autism is very complicated. There'll be a paper coming out in the next 2 weeks from Evan Eichler's group, where they've done full genomic sequencing on about 200 trios, and it's very complicated, extremely complicated. But it's not all genetics. Irva Hertz-Picciotto at UC Davis did a meta-analysis of one of the databases in California, and the upshot of this paper was that, yes, there's increasing prevalence of autism in more recent birth cohorts, especially in the younger age range. When you look at birth cohorts from 1990 to 2003, there's an increased diagnosis of autism in California. And I think this trend has been seen in other states as well. You've all heard about the environment and autism. So you all know what causes autism, right? It's mercury. Obviously, that environmental exposure has dominated the research or the focus of gene by environment interactions in autism. Mercury is a neurotoxicant, there's no doubt about it. And I've said this many times, injecting children at critical periods of postnatal development with mercury, nobody was thinking. Okay, is it causing autism? Is a major driving force for causing autism? The answer is probably not. Not autism core symptoms. Now, children with autism may have adverse effects to mercury because they're more susceptible to the ill effects of mercury, especially when you realize that their immune systems are very different from typical kids. And so we've wondered, how do you approach a complex problem that may be 800 different genes that represent 25% of the cases, 75% of them you really don't know what's causing it, and 86,000 chemicals, of which we've made some attempt through ToxCast and other programs to prioritize. Where do you start? Well, this is where we started. We assumed that environment was, in fact, interplaying with genes that confer risk to autism. And we need to understand the interaction between critical environmental chemicals that alter the neurobiology of the brain in a manner which we understand is changed in autism. That's a slightly different concept. That's a reverse epidemiology approach. We're not going to go look at 10,000 or 80,000 chemicals and do reliable epidemiology with respect to autism. There's just not enough money on the planet. And you're not going to use animals to try to mutate every one of those 800 genes and combinations of those genes to create a mouse that's going to be autistic, because that's not going to happen. There are some optimists that think they have autistic mice, but I don't think you're going to impair the domain of social, well, okay, maybe social, but what about language? I've never heard a mouse speak. So we have gene candidates. We look at environmental chemicals that change brain circuitry and balance in the same way that we know these genes might. And we overlay epigenetics on top of that. And a big part of this is to understand the physiology, which is really, you can enumerate environmental chemicals, you can enumerate genes, but if you don't understand the physiology, the neurobiology of the disorder, I think you're not going to make much headway. So we've taken several approaches at UC Davis. We've done in vitro screening of molecules that disrupt the nervous system. We've used some in vivo animal models, and some of them are really very different from what you've heard before. We've taken antibodies that mothers generate that seem to be a high risk factor and exposed monkeys during gestation to those antibodies. And those results are proving to be extremely informative. We've also adapted that model from non-human primates into mouse. And those are seeming to be very informative. About 15% of mothers have autoantibodies that target brain proteins. We now know what 3 of those proteins are. They're very important in neurodevelopment. Why mom makes antibody titers to fetal brain protein, we don't know. But when we take those antibodies, purify the IgG fraction, and expose animals during critical periods of gestation, the outcome is extremely incriminating that those antibodies are somehow changing the course of the kid's development. And of course, we're using a very strong epidemiological approach, which I will tell you quite a bit about in my remaining time. Okay, so the center is essentially several cores and projects which are interwoven. Our sort of grounding force is the Mind Institute, which has the clinics, which recruits our wonderful participants, which are very eager to help us find solutions. We've collected a huge number of samples in 3 studies. CHARGE, which is a case control study of 2 to 5-year-olds, which now is about 1,300 families from California and regions within California in particular. We recruit through the regional centers. MARBLES, which is a longitudinal study launched off of CHARGE that looks at women who have at least one child with autism and are giving birth to an additional child. So this is a prospective longitudinal study of autism, because the recurrence rate for women that have had an autistic child is about 15%. Okay? There are about 240 women enrolled, and this is over just the last 5 years. It's tremendous. Retention rate's about 90% plus. And then CHARGE back, where we've gone back to the CHARGE participants at a later period in time and retested to see if we could reproduce earlier findings. So this is kind of a chart from the various studies that tap into a longitudinal continuum from conception, pregnancy, through 9 years, 5 to 9 years of age. And Irva Hertz-Picciotto is the PI on the CHARGE study. MARBLES was funded through the center, is now its own PI, R01. And of course, Robin Hansen runs the Mind Clinic, and these are a couple of our participants that consented to having their pictures taken. So what are some of the findings? Well, when we think about the copy number variations that I mentioned earlier that seem to be predominant in terms of risk for autism, there's a difference in how we think about it now. They're not single missense mutation. Those aren't the common ones. You're going to find 20 to 25% of the so-called genetic contributions as copy number variants. And these copy number variants occur within unstable regions of the chromosomes. There are regions of chromosomes that are unstable because, well, as we evolved, they probably contributed our ability to adapt. But that made them unstable. And so maternal age, it's no wonder, is, and paternal age, I'm sorry, I meant parental age, is perhaps one of the larger driving forces for these kinds of copy number variations. Why? Because the mistake occurs during meiosis. And these are stored in the germ cells and then passed on to the child. And so this review really does an up-to-date review on the association between maternal and paternal age and autism risk. Proximity to sources to pesticides. We're now finishing a summary of what the evidence is with respect to autism and pesticides. But really, one of the hallmark papers came out of the state of California Department of Health that indicated that organochlorine, in particular 2 organochlorines, had a contribution of increasing the risk factor about 6-fold, women near fields that were sprayed with 2 organochlorines. Not very, very heavily used organochlorines, but nevertheless still used. And then one which sort of tipped us off, but was never pursued by the author of the first paper, whose name, Roberts, I think, yeah. That of pyrethroids. And there was a risk factor of about 4-fold in women that were near fields or had their homes, no, they were agricultural uses of pyrethroids. And so we pursued this, and we did actually find evidence within CHARGE study of maternal use of pet dips and sprays and an increased autism risk. But we published a paper last year on monoamine oxidase A variant. This is, in fact, a CNV variant. It's a common one. You either have a 3-repeat or a 4-repeat allele. And the 3 repeat allele seemed to contribute risk to autism in our group. Now, this wasn't the first study that showed that, but it was a confirmation that this is a risk factor, a genetic risk factor. But when you put the MAOA C and V with pyrethroid exposures, the risk went up to about 6 to 8-fold. And so this, we think, is an indicator of a gene by environment interaction. That paper was submitted to Nature and then Nature Medicine, and now we're looking elsewhere. Proximity to major highways. This is a study which, in a way, capitalized on early charge location where we did recruitment down in LA. And here, this paper essentially showed that there was about a 1.7 to 2-fold increase in risk of mothers living near highways, major highways, and autism risk. This paper here made the paper of the year at Autism Speaks and also at the NIEHS, where maternal supplementation with folate, especially high supplementation, seemed to confer protection from autism. And I think that's rather significant because here's an environmental factor that's actually an environmental factor that we consider a nutritional supplement. Okay. Another paper that made the paper of the year last year, we were quite busy. This actually was published in 2011, but went online in 2010. Essentially is, if we took a very, very small number of kids that were extremely well phenotyped, with and without autism, all we needed was 10 to get statistical significance to say, you know what? Mitochondrial dysfunction is at a much higher rate in autistic kids than it is in neurotypical kids. Now, some of this data was based on some really interesting work that we capitalized on, that mitochondrial DNA, when energy needs go up, replicate. And so you have mitochondrial DNA replication going on, which increases your oxidative capacity. And this is a natural, very adaptive kind of process. You need more energy, your mitochondrial DNA replicates, and the capacity of each individual mitochondria goes up to provide those needs. However, when something goes wrong, you can have an oxidative stressor, and that can be one of any thousands of oxidative stressors, impact the mitochondria in such a way that you get replication of the mitochondrial DNA, but you have either the same level of oxidative phosphorylation or impaired oxidative phosphorylation. And this is what we found in a large fraction of those kids. Now, not 10 kids, but we now are looking at 300 kids. So we're not sure how this oxidative stress occurs. Oxidative stress is not unique to autism, but maybe this response and something about their mitochondria are in fact unique to autism. So we're still working on that. Okay. Let's just talk very briefly about mercury. Many, many studies and anecdotes, and of course the web, indicate that kids with autism, when their blood's tested for heavy metals, are in the 90 to 98th percentile in terms of mercury burden. We did a case control analysis of this using ICP mass spec of about 400 kids in charge. And I think most of you are aware of the results. Essentially, our children with full-blown autism, those with autism spectrum, have no difference in their distribution of mercury levels at a single time point compared to typically developing kids. And so this says nothing about the contribution of mercury exposure to autism, whether it be neurological or immunological. It just says that kids with mercury, kids with mercury are all diagnostic groups. They're the same. You cannot discriminate amongst the three groups. Our DD group had a little bit lower mercury. Now, the study did find that kids with autism actually had lower mercury, okay, slightly lower mercury. But that's because they don't like fish, and their fish consumption is lower. And so this also pointed out that we sort of indicate that one of the major sources of mercury and a predictor of mercury is what we'd expect, which is fish consumption. Now, we would have stopped there because we also did a mouse model with an immunological susceptibility to autoimmune disorder, the SGL mouse model. That turned out completely negative. And so we did not replicate a previous study that came out of Columbia. But we did look at it a different way. We used global gene expression, transcriptional genomic arrays, to look at if mercury levels predicted similar changes in expression levels in autistic boys and neurotypical boys. And here we used a multiple regression analysis. Frank Sharp and Jeff Gregg essentially found that there were correlations between mercury levels, because we had a huge distribution of mercury levels in these kids, did define certain genes that were either up or down regulated, but surprisingly, they weren't the same genes in the two diagnostic groups. In the autistic group, some genes went up when those same genes didn't change in the neurotypical, and other genes went down where they went up in the neurotypical. And so one weakness of this study is that, in fact, multiple comparisons couldn't be done. So we basically just picked genes that were in common, but changed with mercury at an alpha of less than 0.05. So there is some possibility that some of these findings were serendipitous. So here are some of the conclusions. I'm going to go on because more conclusions. Okay. PBDEs. Our kids in charge have some of the highest PBDE levels that have been reported. They're 2 to 5 years old, and their their PBDE levels are probably 5 times higher than kids on the East Coast, and about 10 times higher than those in Nordic countries. PBDEs have a very different effect on immune cells from autistic kids than they do on neurotypical kids. It's a small pilot study that we published, and now we've replicated it and are going to publish a larger study. This is with Judy Vanderwater and Paul Ashwood, who are immunologists. So, how do we get at using useful models for screening when you have such a complex disorder? You can use animal models. Some of them will be disappointing. Others may suggest that social impairments occur. Cells will tell you whether you're affecting similar pathways in a mouse compared to what you see neurologically in children. But it's self-limiting. So we took a different strategy. Oh, yeah, this is part of the strategy. We took 600 kids, isolated DNA, 300 of them, approximately 300 of them were full-blown autism, 300 of them were neurotypical. The demographics is a population-based demographic that's not all that different from California demographics. And I want to point out that 88% of DNA in the Simons Foundation and in other collections are Caucasian. So this is a more diverse, representative population from which to collect DNA. And this hotspot 1 array is an array that Evan Eichler designed that looks specifically at regions that are unstable within chromosomes. And here now you have 130 plus regions that are targeted by the probes on these arrays. So I think our data set is quite unique in the sense that it's not 90% Caucasian and 3% Hispanic, and other ethnic groups are underrepresented, which is what the current state of affairs are in autism research, but in fact population-based. What do you look for? These probes, we started this work actually 5 years ago when I gave a talk at the University of Minnesota and ran into Scott Selleck, who came up with this idea, said that the GWAS studies and SNP studies, common variant studies, currently, or at that point in time, seemed very exciting, but were not going to pan out. And in fact, he was right. His view was, as Evan Eichler's view, although Evan was not working on autism at the time, was we were looking in the wrong place because most of the arrays that were being used essentially ignore regions of genomic instability because it's very hard to lay down probes in these regions. Unless you design them in such a way that you're actually targeting those regions. And so we used two types of arrays. One was a custom array from Nimblegen that Scott designed, and then later the discovery array, the hotspot 1 array from Evan Eichler, where now we're up to about 600 kids that we have this kind of information on. I can't summarize it all, but we found many unique pathogenic CNVs in the CHARGE study. But I think what's most important is that the copy number burden in autism is much higher than the copy number burden in typically developing kids. So they just have more copy number variants in their genome than do kids that are typically developing. And as you can see here, as would be expected, kids with intellectual disability, which is a broad array of disorders, also have a very high copy number burden. Now, what are some of these variants? Are they ones that you would expect that people have already published on? Well, here's one that just blew us away. This is a trio that I'm going to show you. This is chromosome 4, and the index child is extremely autistic. The red here is a segment of chromosome 4 that has been deleted. It's not 100 kilobases. It's 3.5 megabase deletion. This is a huge, what we call a microdeletion, okay? It's huge. A geneticist would look at this, compare it to the known databases. If this was unique, never found in a typical genome, this would be considered essentially a causative mutation. So far, so good, right? Mom did not pass this deletion onto the child. She doesn't have the deletion. Dad passed it. So this was paternally transmitted to the child. As far as we know, Dad may have some issues, but is not autistic. So this raises several important questions. That this deletion is not sufficient to cause the child's autism, and in fact may be setting up the child for second and third hits, whether they be genes that come in through mom outside this region, or an environmental insult that the child received either during development or postnatally. So this deletion is not observed in about 16,000 developmentally delayed individuals that have been screened, or about 8,300 neurotypical children. So this is very interesting. One of the things we want to do is take fibroblasts from these children, from this family. We're going to be very specific here because it's not easy to do. Reprogram their fibroblasts to neural cells and study the neurobiology by which the index child, the mother, and the father, where they divert in neurodevelopment. If we can understand this, then we can use these cells to understand environmental susceptibility. Okay, so I want to give you, and just finish here, some of the work that we've done to reach that end. We weren't going to start with this rare CNV, this rare deletion. We decided what we were going to do, since Fragile X syndrome, and we have some of the top-notch Fragile X researchers at UC Davis, the Hagermans, we decided that we were going to target Fragile X syndrome as our first reprogramming attempt. And there's a lot of benefit there. There's a 20% autism rate, and the size of the repeat within the FMR1 gene can vary from normal to 8 to 10 repeats, to a gray zone, which is about 15 to 30, 40 repeats, to the pre-mutation, which is causative for a late onset set of disorders, which is somewhere between 100 and 200, and then Fragile X syndrome, which is over 200 repeats, which causes the gene to inactivate. So Fragile X is a neurodevelopmental disorder. FASTAS, Fragile X associated tremor ataxia syndrome, is the same gene. It's just how many repeats, duplication within the gene of a CGG trinucleotide repeat you have. And so we were fortunate enough to find an individual that was in the pre-mutation range, okay, didn't have Fragile X, and actually hadn't been diagnosed with FASTAS at this point, but clearly had a pre-mutation, so 130 repeats on one X chromosome, female. The other was in the normal range. And it so happens that you can use cloning and subcloning of fibroblasts to isolate cells where each of the X chromosomes are stable, the one that carries the mutation, the one that doesn't. This is extremely important because the rest of the genome is identical. So you're getting a mutant cell and a normal cell from the same individual, same genetic background, and now you're going to say, I'm going to reprogram those cells to an embryonic stem cell, an iPSC, and then you're going to drive them forward to a neural differentiation. And essentially, this is what we did here, is these are about 3 weeks out from the post-differentiation process, and you can see these cells are now extending neurites, and they have positive staining for neural markers. And unfortunately, I'm not going to be able to show you this because I don't think this, does it work? These cells are very active signaling. You can see them light up, okay? And we can measure functional differences between the normal and those cells, those neurons that carry the mutation. They tend to be hyperexcitable, which is consistent with what we know about this disorder. So this is the normal. This is the one carrying the mutation. These are cells carrying the mutation. These signals that we're measuring here are calcium signals within these cells. But these signals are essentially mediated through excitability of the cells because we can block them with tetrodotoxin, which blocks action potentials. The other thing that we found that was very, very interesting was that their response to glutamate is quite different. The black response here, the curve, is normal cells. The red is the cells carrying the mutation. And an imbalance in excitatory-inhibitory signal has been proposed for these kinds of mutations in at least Fragile X. Okay? This recapitulates our mouse model. So these are mouse neurons now, normal, carrying the pre-mutation. You could see that the excitability of the cells are quite different and mirror those of the human cell. So I think I'm going to stop here because, and this is sort of our mantra, that there are many mutations, copy number variants, and probably methylation issues that confer heritable risk to autism. In many cases, repair mechanisms probably mitigate those problems, but not completely, because autism is a spectrum of presentations. Environmental exposures, either directly on the genome itself through these unstable regions of the chromosome, or through signaling systems that are participating in contributing risks, influence the clinical phenotype. It'll probably be very difficult to ever find a single chemical that causes autism. So we're thinking that what we really should be looking at is environmental chemicals that can promote the progression of, let's say, a mild case of autism to a severe case of autism, where if you look at the tail of that distribution where you might have 5% that would be very much on the margin and maybe not autistic and could be given a lot of help to overcome their autism, that environmental exposures may take them over that edge. And so that's kind of where I'd like to leave it off. Lots of people to thank. At UC Davis, I missed Evan Eichler's group here and Scott Selleck's group because I actually was meeting with Evan last night, and I didn't get home till very late, and I didn't have a chance to modify the slide. I'd be happy to answer any questions.